Hello, dear students. Namaskar. Now we are going to discuss about electron transport chain. This electron transport chain is known by a lot of names. There is one biological oxidation, cellular respiration, electron transport chain, oxidative phosphorylation, like that. So let us define electron transport chain. The definition of electron transport chain, it is from that statement itself, it is clear. Electrons are transported through a chain. Electrons are transported through a chain. So where this chain is situated? This chain is situated in the inner surface of the inner membrane of the mitochondrion. You know the structure of mitochondrion. Mitochondrion is having a matrix, and outer to that matrix there are two membranes, inner membrane and outer membrane. There is a space in between this inner and outer membrane. So these spaces are very significant in energy transactions. So this chain is situated in the inner surface of the inner membrane of mitochondria. And this chain units, for a chain there are units. These units are linked together like that, chain. So what are these units? These units are nothing but enzyme complexes. Enzyme complexes made of protein. So these units are protein enzyme complexes linked together by coenzymes. Coenzyme Q and cytochrome C. So these are the connecting links between this chain units. So that is the physical structure of electron transport chain. So through this, electrons are transported. From where these electrons comes? So let us think about that. These electrons are nothing but, as I told you, in Krebs cycle, we are generating the solar energy installed hydrogen electrons are captured in the form of NADS2, FADS2. NADS2, FADS2. These are reducing equivalents. They are energized reducing equivalents. So these reducing equivalents are channeled to this electron transport chain. Okay, so these are the electrons. So these electrons means hydrogen electrons, hydrogen ions with electrons in the outer orbit. These electrons are charged with solar energy. I told you in the Krebs cycle, this happens at the time of photosynthesis. So that energy is captured through these various processes. So the final end of metabolisms, final end of the metabolism energy transaction is electron transport chain. So with the electron transport chain, the whole energy which is present in the food is transferred from the food to our body cells, to our body systems, to our body activities in the form of ATP. So ATPs are known as cellular batteries. So into that we are installing this energy of sun. So that energy can be easily used by the cells for its various purposes. So that is electron transport chain, through which electrons, this hydrogen electron, solar hydrogen electrons are transferred and energy is captured in the form of ATP. So this is the physical structure of electron transport chain. Now, before going through the electron transport chain in detail, we have to know certain concepts of energy transaction. There are two types of energy which are utilized in the body. That is substrate level phosphorylation energy and oxidative phosphorylation energy. One group of energy is substrate level phosphorylation energy. Other group is known as oxidative phosphorylation energy. In both these groups, phosphates groups are used to transfer this energy. Why? Because phosphate is having valency 3. So 3 hydrogen electrons can get accommodated in the phosphate. That is why phosphate is used. So, what is substrate level phosphorylation? Substrate level phosphorylation is the energy source which is away, which is not related with electron transport chain. That is substrate level, which is not related with electron transport chain. That is substrate level. Example, you have seen this substrate level phosphorylation in glycolysis, aerobic glycolysis. We have seen that pyruvate kinase, one phosphate is formed, ATP is formed. That is substrate level phosphorylation. Then bisphosphoglycerate kinase, that is substrate level phosphorylation. In Krebs cycle, we have seen that succinyl thiokinase, their GTP is formed. So these are all substrate level phosphorylations which are not related with electron transport chain. Another group of energy transaction is oxidative phosphorylation. That is nothing but electron transport chain phosphorylation. Electron transport chain phosphorylation. So oxidative phosphorylation is taking place inside the mitochondria. But the other substrate level phosphorylation usually in cytoplasm, or need not be in mitochondria. Especially this glycolysis that happens in cytoplasm. So substrate level phosphorylation is not that much related with the mitochondria, but oxidative phosphorylation is totally related with mitochondria. So these are two types of phosphorylations which are important in case of energy transaction. So this will be asked as a short note for 1 mark or 2 mark. The question will be like this: differentiate between substrate level phosphorylation and oxidative phosphorylation. So you have to mention these points. And you have to mention the examples of substrate level phosphorylation also. Now, before entering into the electron transport chain, we have to know about the high energy compounds, because actually electron transport chain provides only ATP, but certain high energy compounds are there which on which the body depends for cellular activities, metabolic activities, biochemical activities. So for example, of course ATP is related directly with GTP, ATP, CTP, all these are directly related with carbohydrate metabolism. Like that, protein metabolism high energy compounds are there. That is SAM, you know that SAM, acetylanosyl methionine, creatine phosphate. All these are protein related high energy compounds. And fat related high energy compounds like acetyl coenzyme, succinyl coenzyme. So you have to study that also. It is given as a table in all the textbooks, especially in Vasudevan sir's textbook. You can see the table which mention almost all the high energy compounds as a table. So you study that. That is also important because sometimes they will ask, for 1 mark, they will ask the question like this: mention 2 high energy compounds originated from protein metabolism. So you can answer that. Now, before entering into the electron transport chain, another important point, which are usually asked as short note for 3 marks or 4 marks or 5 marks, that are 2 shuttles are there. What do you mean by shuttle? Shuttle is nothing but we are transporting something from some place to another place. Here, this shuttling occurs from cytoplasm to mitochondria. So 2 shuttles are important here, because this reducing equivalence, NADH2, is not permeable to the mitochondrial membrane. Reducing equivalence, NADH2, is not permeable through the mitochondrial membrane. So they cannot be directly transferred from cytoplasm to mitochondria, especially the things that are coming from the glycolysis. Glyceraldehyde 3-phosphate, dehydrogenase enzyme, they brings a lot of NADH2. So those NADH2 cannot be directly transferred from cytoplasm to mitochondria, because this mitochondrial membrane is impermeable to NADH2. So by some messengers, by some carriers, we have to transport this NADH2 to the mitochondria. So which are the carriers? Here we have two major molecules. That is, one is malate. Malate is used as a transporter or a shuttling agent. So that is known as malate shuttle 1. Another is glycerol 3-phosphate shuttle. So these two shuttles are there to transport NADH2 from cytoplasm to mitochondrion. So what is malate shuttle? Here, initially what happens? In the cytoplasm, what happens? Malate will be attached with this electrons of NADH2. So one molecule of NADH2 will approach the malate. So that hydrogen electrons have to be transferred to malate. So with that, oxaloacetate is used. So oxaloacetate will be dehydrogenated to malate by attaching with this electrons. So the oxaloacetate will become malate. Malate is easily permeable through the mitochondrial membrane. So the malate will enters into mitochondria easily. After reaching into the mitochondrium, it will donate or it will release the accepted electrons and it will again convert it back to oxaloacetate. So one NAD will be waiting there to accept these electrons. So the NAD will become NADH2. Understand? So that is how this NADH2 is transferred from cytoplasm to mitochondria. Understand? Then after releasing these electrons, this malate will become oxaloacetate. So that oxaloacetate has to be transferred out. So oxaloacetate is not that much permeable as malate. So with the help of another amino acid, this oxaloacetate will be transaminated. That amino acid is glutamate. So glutamate will transaminate oxaloacetate to aspartic acid. And the glutamate will become alpha ketoglutarate. This alpha ketoglutarate and aspartate both are easily permeable through the mitochondrial membrane. So they will leak out into the cytoplasm. After reaching the cytoplasm, again this oxaloacetate will be formed from the aspartate with the help of alpha ketoglutarate by this transamination process. And at the end, oxaloacetate is formed, alpha ketoglutarate is formed. And glutamate is also formed. Glutamate is also formed. Understand? So this glutamate will be leaked into the mitochondrion. The diagram is given there. I'll give you the PowerPoint. In that beautiful diagram is given there. So you can go through that diagram. In all the textbook, this malate shuttle diagram is there. So it's clearly explain the process, what is really happening there. The purpose of this shuttle is this, because directly NADH2 cannot be transported through the mitochondrial membrane. It has to be transported through a medium of malate, which is formed from oxaloacetate by dehydrogenation process by the enzyme malate dehydrogenase. Okay, so this is the malate shuttle. Same is the case of glycerol 3-phosphate shuttle. Here, same as this NADH2 will hydrogenate dihydroxyacetone phosphate, and the dihydroxyacetone phosphate will become glycerol 3-phosphate. And this glycerol phosphate, which is carrying these electrons, high energy electrons, that will be easily permeable through the mitochondrial membrane, and that enters into the mitochondrial membrane. And after reaching the mitochondrium, it will release that electrons, and it will again become dihydroxyacetone phosphate. And there it is not NAD accepting this electron, but FAD is there to accept this electron. So by the glycerol 3-phosphate shuttle, inside mitochondrion, FADH2 is formed instead of NADH2. So the electrons are carried through the glycerol 3-phosphate, but they are donated to FAD. Only thing is that if it was NAD, we could have make 3 ATP, but it is FADH2 because because of that we can make only 2 ATP molecules. So anyhow, these two shuttles, the diagrams are given in the textbooks. You have to draw the diagrams very well, and these shuttles will be asked as short note, 3 marks or 4 marks. So study that very well. Now, we will see the sequence of electron transport chain. Now, what is the substrate we have before starting the electron transport chain? The NADS2 coming from the NADS2 reducing equivalents coming from glycolysis and citric acid cycle, and FADS2 that is coming from citric acid cycle. All these are coming, and they are available now in the mitochondrion and in the inner membrane of the inner surface of the inner membrane of the mitochondrion. They will pass through various complexes. I told these complexes are protein complexes, protein enzyme complexes, because this is a combination of a lot of chemical biomolecules. So that is why they are known as complex. So we are designating them as complex 1, complex 2, complex 3, complex 4. And these complexes are connected together by coenzyme Q, cytochrome C, like that. So NADH2 group of electrons will enter directly through the complex 1 enzymes, and they will pass through the coenzyme Q and complex 3 and complex 4. You can see the diagram. While you are studying this, you have to see the diagram. You have to draw this diagram and make it by heart. It's very important. So, how they are generating this ATP? See, while this, NADS2 is a high energy compound charged with hydrogen electrons, solar hydrogen electrons. So just like a bullet, it pierces this complex. And when it is released from that complex, just like a bullet released from the barrel of a gun, there will be spark, energy, there will be fire. You can say when somebody is using a gun, when they fire, there will be firing and spark. Because these bullets are released with the maximum velocity, triggered out. The triggering out of the bullet. Same is happening there. There will be an energy release. So this energy release will cause a lot of configurational structural changes in the protein environment. As a part of that, there will be a difference in the electrochemical potential gradient across the inner membrane of the mitochondria. So as a result of that, so many of these electrons, hydrogen electrons, will be escape into the intermembrane space of the electron transport chain. Understand? So they will be charged up. Again, this bullet is next passing through the coenzyme Q and complex 3. Next is complex 3. So when it passes through and pierces the complex 3, again there will be energy release. With that energy release, there will be changes in the electrochemical potential gradient, and again so many electric hydrogen will be escaped into the intermembrane space. Like that, by the passage of these NADS2 bullets. So you have to study various chemical changes that happens in each complexes. It is very complicated thing, but simply it is given inside the textbook. You have to study that. What actually really happens? Nothing but transaction of these electrons. I can compare that with a 4 into 400 meters relay. So when 4 people are actually running, but they are running with a baton. The first person will run and hand over this baton to the second person. The second person, with that baton, he will run and he will hand over that to third person. Third person will give to fourth person, like that 4 into 400 meters relay. Here, instead of baton, these electrons, the solar electrons. Understand? So, same is the case of FADS2. But FADS2 entering through the complex 2 only. So, it misses the complex 1. That is why it is having 2 pairs. It will pierce the complex 3 and complex 4. But the NADS2 will pierce the complex 1, complex 3 and complex 4. So, it will generate 3 ATP, but this one will generate 2 ATP. Why? Because along with this piercing and changes in this electrochemical potential gradient, a corresponding ATP is synthesized through the ATP synthase enzyme molecule. So that is why this NADH2 creates 3 ATP, FADH2 creates 2 ATP. So this is how this electrons, when they pass through these enzyme complexes, they will generate energy. This generate energy is a heat energy. So because of this energy, our body is warm. Because of this energy, after the metabolism, the whole cellular world become maintaining that body temperature. Understand? So that is about, so to understand it, see, whenever any subject is interacting with another subject, there will be a release of energy. So for example, if you are scratching a rock with a knife, what will happen? There will be a spark of fire. Same is the case. When these electrons or bullets are passing through the same complexes, there will be a spark. But that spark will make a lot of changes in the permeability of the membrane. Understand? Okay. So that is the mechanism of electron transport. Now, you have to study about the ATP synthase enzyme complex. ATP synthase enzyme complex. What is ATP synthase enzyme complex? It is nothing but an enzyme complex, or just like a tube which is situated in the inner membrane of the mitochondria. See, with the piercing of these bullets, piercing of these electrons through the enzyme complexes, I told that there will be leak of hydrogen electrons into the space, hydrogen ions, protons into the space, intermembrane space. So whenever this more and more electrons are escaped into that space, there will be a change in the electrochemical gradient. So this will create an osmotic gradient. An osmotic gradient. The ionic concentration will increase in the intermembrane space. So the extra things which are accumulated there will be gushed through the holes which are present in this ATP synthase enzyme complex. Just like, you just imagine about a hydroelectric project. In that, water is there, and this water is gushed through a turbine of motor. So with the gush of water, with the power of water, we are generating energy. Same is happening here. Instead of water, these hydrogen electrons are flowing through this ATP synthase complex. Understand? So that power is captured in the form of ATP. ADP will be converted to ATP. What is the structure of this ATP synthase enzyme complex? It is having two subunits. One is F1 and 2 is F0. F1 and F0. This F0 is mainly embedded in the membrane, and F1 is projected to that intermembrane space. So through the hole, these electrons are traveling into the matrix of the mitochondria. Okay, I told what is the driving force because of the difference in the electrochemical potential gradient across the membrane. That is how they are gushed into. If water is there, high amount of water is somewhere, low amount of water is otherwhere, so naturally with the gravity, the water will flow. Same is the thing happening there. Okay, but this F1 and F0 are, F1 is just like a ball, and F0 is just like a rod. But there is a hole inside. And if this F1 is composed of mainly alpha and beta subunits. 3 alpha beta subunits are there, 3 alpha subunits are there, and 3 beta subunits are there. They are arranged like the petals of a flower, and there will be configurational changes, structural changes while these electrons are passing through. Same, but this F0 thing is having a rod like a tube like structure. So when this electron pass, it will rotate. So you know, when we are firing with a pistol, we will rotate it. The bullets are rotated, then they are firing. Same is the case. So this, whenever there is a energy generated by passing, that energy will be captured by the ADP to ATP. So energy is traveling on phosphate. Why phosphate is selected? I thought that phosphate is having maximum hydrogen electron carrying capacity. So ATP is generated like that. So this is the process of ATP synthesis. And at the end, what is the driving force of these bullets from complex 1 to complex 4? That is known as, that driving force of this reducing equivalent electron bullets are known as a redox potential. Because at the left end it is negatively charged, at the right end it is positively charged. Who is giving this positive charge at the right end? Oxygen. The oxygen that we inspire. The oxygen that we inspire make this redox potential. That is a driving force for these electrons who are attracted. The maximum power of attraction in this universe is between hydrogen and oxygen. Hydrogen attracts oxygen. Oxygen attracts hydrogen. That is known as a redox potential. So this extra hydrogen electrons released will be accepted by this oxygen and form water. So at the end, water is formed. I told in your Krebs cycle, citric acid cycle, this separation of water takes place at the beginning of the photosynthesis and it ends at the end of electron transport chain. So water is split there, water is formed here. So the story of energy begin with the split of water by sun and ends with the union of hydrogen and oxygen to form water by the same sun. Understand? So sun is a master of our life. Why he is the master of life? Because he is the provider of energy. He is the he gives maintenance of energy. He is the witness for all energy transactions. Now, we have to think about uncouplers. Is a very important term used in electron transport chain. What are uncouplers? This will be asked as a 2-mark question or 1-mark question. Sometimes they will ask like this: name an uncoupler or name 2 uncouplers. What is the difference between physiological uncouplers and chemical uncouplers? What is uncoupler? Uncoupler is nothing but the word itself. It's uncouples. It's diverse. You know, divorce. Husband and wife will divorce like that. Uncoupling, that is also an uncoupling. Here, uncoupling takes place between oxidation and phosphorylation. Actually, oxidation and phosphorylations are linked together, always coupled. With the oxidation, phosphorylation takes place. With the oxidation, phosphorylation takes place. With the oxidation, passage of these bullets of reducing equivalents, the ATP generation takes place. That is linked together. So uncouplers will dealing this coupling. So what will happen? This oxidation process will take place, but ATP generation will be prevented. But what at that time what happened? There will be a lot of energy released as heat. Because energy will be released as heat. So this is the function of physiological uncouplers. Physiological uncouplers. Very important physiological uncoupler is thermogenin. Thermogenin is a protein which is present in the brown adipose tissues of newborn mammals. These mammals, when they are born, they have to maintain their body temperature. They have to adjust with the body temperature of the environment. So heat generation is more in the adipose tissues of the newborn mammals. So there the thermogenin is acting as a physiological uncoupler which will prevent the phosphorylation, but it will promote the oxidation. So the body will become warm. Understand? And other physiological uncouplers, mainly thyroid hormone is also active. Hypothyroidism, thyroid hormone is also a physiological uncoupler. By the maintaining the metabolism, the body temperature will also be maintained. So that is also having an uncoupling like activity. And chemical uncouplers are there: dinitrophenol, dinitrocresol. So all those things are present in a disinfectant wash, detergents, no. So they will kill the microbes by this by uncoupling their mitochondrial oxidative phosphorylation. So those things are also given as a table in the textbooks. So you have to study that name two or three uncouplers name. That's enough. And physiological uncoupling is very important. Thermogenin is very important. That will be asked as a 1 mark question. But you have to understand what is the concept of uncoupling. Uncoupling is nothing but separating the oxidative process, redox potential driving force from the phosphorylation process. That is uncoupling, separation, delinking the oxidation from phosphorylation. Understand? That is uncoupling. And we have to study about the inhibitants of oxidative phosphorylation or electron transport chain. So inhibitants means inhibitants of this ETC are two types. Those who inhibit this electron transport of oxidation traveling process, and other group, those who inhibit the ATP synthase enzyme complex. Generation of ATP is inhibited. There are the major two types of inhibitors. One is the inhibitors which act on these complexes. The other group which is act on the ATP synthase. Understand? So you have to study 2 or 3 examples of these, both groups of these inhibitors. These electron transport inhibitors are cyanide, hydrogen sulfide, carbon monoxide. They will inhibit the passage of electrons through the complexes. They will inhibit each and every complexes. So the details of that is also given as a table in the textbooks. You can study that. And ATP synthase inhibitors are there. Certain antibiotics are acting. Certain antibiotics are acting as inhibitors. They will kill the bacteria by inhibiting the ATP synthesis. They will inhibit the ATP synthesis of the microbial organism, and thus they kill. Valinomycin is an example of such an antibiotic which will inhibit the ATP synthesis of the bacteria. So with this, we come to the conclusion of the electron transport chain. So I will just narrate once again. When you study the electron transport chain, you have to think the following points. First, you have to know what is electron transport chain, the concept of electron transport chain, how energy is generated from electron transport chain by the process of oxidative phosphorylation. What is oxidation? The process of transfer of these electrons across the complexes, that is oxidation, with the driving force of redox potential created by oxygen at the right end. Okay, that is electron transport chain. Next, you have to know about how this NADH2 are transported from cytoplasm to mitochondria by these two shuttles, malate shuttle and glycerol 3-phosphate shuttle. Glycerol 3-phosphate shuttle, different from malate shuttle, is by the acceptance inside the mitochondrion is by FAD, not NAD. Okay, that is another important point. You have to know the sequence of this electron transport chain: complex 1, coenzyme Q, complex 2, complex 3, complex 4, like that. Okay, that diagram is very important. When this reducing equivalents are piercing, this complex's energy will be released and this hydrogen electrons will be escaped into the intermembrane space. That you have to understand. That is the sequence. Next, you have to know what is uncoupler. What is being uncoupled? The oxidation is being uncoupled from phosphorylation, or delinking oxidation from phosphorylation. So which are the things which delink? Physiological and chemical. And inhibitors. Inhibitors are also two types. The inhibitors which inhibit this transport of electrons through these complexes, and the inhibitors which inhibit the ATP synthase enzyme. And another important, you have to know about a basic structure of the ATP synthase enzyme complex. It is made of F1 and F0 unit. F1 is made of alpha beta, F0 is made of gamma. There's a hole through all this. When these electrons are flowing, just like what happens in a hydroelectric project, how this electricity is generated from water, same way ATP is generated from electrons. Understand? So that's all about the electron transport chain. Study that well. All the best.